Okay, so welcome to this next video in which we're talking about the 5-HT3 receptors and their involvement in depression. Okay, so we've now seen the structure of 5-HT itself, or serotonin as its other name is. Now what we want to see is the structure of these 5-HT3 receptors. So basically, there are 7 families of receptors for 5-HT, and these are the 5-HT1 receptors all the way through to the 5-HT7 receptors. Okay, 5-HT7. Okay, so let me make it clear, these are not 7 receptors, instead these are 7 families of receptors. So you have the 5-HT1 family of 5-HT receptors, the 5-HT2 family of receptors, the 5-HT3 family of receptors. We are looking at the 5-HT3 family of receptors. Okay, and they are the only receptors for serotonin, for 5-HT, which are ligand-gated ion channels, LGICs for short. Okay, and specifically they are what are known as cis-loop ligand-gated ion channels. So I will now explain to you the structure of these 5-HT3 receptors. And there's more than one. There's not just one 5-HT3 receptor. As I say, it's a family of receptors. And it's the family of serotonin receptors which are ligand-gated ion channels, as opposed to G-protein coupled receptors, which all the other families of 5-HT3 receptors are. Okay, so if we have the membrane here, so this is the phospholipid bilayer here, and then we have our 5-HT3 receptor sitting here. Now it's a ligand-gated ion channel, so I need to draw it with a pore down the middle, and specifically it's a cationic channel, so it allows monovalent cations such as sodium and potassium to move through it. Okay, so here's the phospholipid bilayer on both sides here. Here's the pore through which the ions are going to move, and the 5-HT3 receptors, whichever one we're talking about, they are not just one protein. Instead, this ion channel is made up of 5 separate proteins. So I'll divide it up. You have 1, 2, 3, 4, 5 separate proteins which all join together like so to make the 5-HT3 receptor. Okay, so there are these 5 separate proteins all coming together to make the full receptor. So this is a 5-HT3 receptor. Okay, right, so now what we're going to do is we're going to pull out one of these individual subunits here. Okay, one of these individual subunits within the pentameric whole receptor. And we're going to look at the structure of this individual subunit, i.e. how it spans the membrane. So, by the way, this is a pentamer. Okay, so a pentamer just means something made up of 5 constituent portions. So, we're now going to pull out this subunit of the 5-HT3 receptor pentamer and have a look at its membrane spanning topology. And by the way, this is the extracellular side here, although it doesn't really matter for this drawing, but this is the extracellular fluid, and this is the cytoplasm. Okay, and we'll stick to that convention in this next drawing where it will matter. Okay, so here's the phospholipid bilayer. Okay, this is the ECF, or extracellular fluid, and this is the cytoplasmic side. Okay, and the structure of one of these individual subunits of the 5-HT3 receptor looks like this. You have the amino terminus here, so here's the amino terminus. Then you have something in the extracellular domain known as a cis loop, which is a loop in the polypeptide structure. So remember, this is a polymer of amino acids joined one after the other. And you have this loop in the polypeptide structure which is held together by disulfide bonds between 2 cysteine amino acids, and that's the cis loop. So these are called cis-loop ligand-gated ion channels because the subunits of this pentamer all have this cis-loop structure in their extracellular domain. Okay, then after the cis loop, what happens is you have the first membrane spanning alpha helix, the second membrane-spanning alpha helix, the third membrane-spanning alpha helix, a large intracellular loop known as the intracellular loop, or also as the M3-M4 loop, and then the fourth membrane-spanning alpha helix, and then the C-terminus of the polypeptide. Okay, so let me highlight some important portions of this structure. So we have these 4 membrane-spanning alpha helices here, shown in turquoise, and these are labelled M1 through M4. Okay, so these are the membrane-spanning alpha helices. This first one here, which is closest to the amino terminus, that's the M1 membrane-spanning alpha helix. The second one is the M2 membrane-spanning alpha helix. Then the third one, M3, and the fourth one, M4. Okay, and those are that form the transmembrane region of each of these subunits of the 5-HT3 receptor. Okay, you then have, between M3 and M4, a large intracellular loop here in blue. Okay, so this is the intracellular loop, or also called the M3-M4 loop. Okay, but this is also known as the intracellular loop. Intracellular loop. And this makes up the intracellular domain of the 5-HT3 receptor. Okay, now this large extracellular portion over here, and I've completely over-exaggerated this. The way I've drawn this, I've had the cis loop completely dominating the extracellular domain. So this entire orange bit here, this pre-M1 region, this is the extracellular or EC domain. Now, in my picture, it is dominated by this cis loop that I've drawn here. So this is the cis loop. In reality, the extracellular domain contains a lot more than just the cis loop, but the cis loop I've emphasized so that you understand why it's referred to as a cis loop ligand-gated ion channel. Okay, so I'll just discuss with you what a cis loop actually is to make sure that we're all completely familiar with that concept. So it's basically just a loop in the polypeptide structure. So here is the polypeptide. So I'm drawing a line because I'm too lazy to draw amino acid polymerized to amino acid polymerized to amino acid, but that's what you would have. Let's say this is an amino acid, then here's the next amino acid, the next amino acid, the next amino acid, and it would go on. And then suddenly you'd have a cysteine amino acid, and this one is going to be drawn completely out of proportion. So I'm actually going to draw this amino acid rather than just having a tiny little portion of a line to represent it. I'm actually going to draw it out. So it's going to dominate, it's going to look massive compared to the others, but that can't be helped. So here's the alpha carbon here with the methylene group coming off, and then cysteine amino acids have a methylene group with then a thiol group bound to it. However, I'm not drawing the full thiol group. A thiol group is actually a sulfur bound to a hydrogen, so this is a thiol group. So it's similar actually to an alcohol group, but instead of oxygen here you have sulfur. So this is a thiol group. So cysteine amino acids would have a thiol group here. However, I'm being fortuitous. I know that this thiol group is going to be part of a disulfide bond, so I'm not actually drawing the hydrogen on because it will have come off. Okay, so here's the cysteine amino acid, and then the polypeptide continues. So again, I'm not drawing the individual amino acids here, I'm just drawing a line to represent the polymer of amino acids. Okay, and then you'll have another amino acid here. So here's the amino group, here's the alpha carbon, here's the methylene group here off the alpha carbon, and then you would have a thiol group off here as well. So this is another cysteine amino acid. However, again, I know this is going to be part of this disulfide bond between the two, so I'm not drawing the hydrogen. So on goes the polypeptide. Oops. Okay, right. So you then have this bond between the two sulfurs, and this is the essential bond. This is what's holding together the cis loop. This. Okay, so this is a disulfide bond, also known as a disulfide bridge. So disulfide bond, also called a disulfide bridge. Okay, and it's between these two cysteine amino acids which are on opposing strands. So this is a cysteine amino acid. Okay, and the 3-letter amino acid code for cysteine is CYS, cis. So this is why this is called a cis loop. It is a loop in the polypeptide structure where it's being held together by disulfide bonds between two cysteine amino acids on opposing sides of the loop, basically. And that's why it's called a cis loop. Okay, so that's the structure then of a 5-HT3 receptor. You have 5 of these 5-HT receptor subunits coming together to make this pentamer, which is then a full 5-HT receptor, or a 5-HT3 receptor. Right, now the simple thing, the simple ideal world, in the simple ideal world, there will be one gene coding for the subunit of a 5-HT3 receptor. It would be called the 5-HT3 receptor subunit gene, and you would just use this gene to make 5 copies of the protein, and you'd stick those 5 identical copies of the protein together to make a 5-HT receptor. However, the world is not ideal. Instead, we have 5 cloned genes for 5-HT3 receptor subunits, okay? So there isn't just one gene coding for a 5-HT3 receptor subunit. Instead, there are 5 genes, and these are called, okay, so they are called the 5-HT3A gene, okay, the 5-HT3B gene, and then it goes on, and I might as well finish it now. Only 3 more. 5-HT3C gene, and then 5-HT3D and 5-HT3E. Okay, 5-HT3D, and then 5-HT3E. Right, so all of these genes will have slightly different sequences of slightly different sequences of organic bases in the DNA, and that will lead to the proteins they produce having slightly different sequences of amino acids. However, the proteins that all 5 of them produce are similar enough that they have the same membrane-spanning topology in the membrane, so they form this same structure that I've shown you. So here's the phospholipid bilayer. Here's the amino group of this protein, and then it forms the cis loop here, and then it forms M1, M2, M3, the M3, M4 loop, and then M4, and then the C terminus like that. So they all have the same membrane-spanning topology here. Okay. However, they do all have slightly different primary amino acid structures, and they do therefore all have slightly different properties. So we have 5 subunits of the 5-HT3 receptor: the 5-HT3A receptor subunit, the 5-HT3B receptor subunit, and so on. So the question then becomes, how do we assemble these into pentamers, basically? Now, there's some good news. There are only 3, only 2 5-HT3 receptors which have actually been shown to be non-trivial in human physiology. The others may well have some role somewhere, some little convoluted role, but they haven't been characterized yet. So basically, there are only 2 5-HT3 receptors that you need to worry about. So what are these 2? Well, basically, if I draw a picture where we're looking down at the receptor from above, so I'll draw one of these cartwheel style pictures. So here's the ion channel going down. Here are the 5 subunits making up the 5-HT3 receptor. Now, basically, one of the 5-HT3 receptors that you find is what's known as the 5-HT3A homopentamer. Okay? Homo means the same. So basically, in this homopentamer, what you do is you have 5 identical subunits in all 5 slots of the 5-HT3 receptor. So in this slot here, you have a 5-HT3A subunit. In this slot, you also have a 5-HT3A subunit. So basically, absolutely all of them are the 5-HT3A receptor subunit, and I can't fit in the full name of it, so I'll just put 3A in each of these. So you go to the 5-HT3A gene, you use it 5 times to make 5 identical proteins, and you stick these together to make a 5-HT3A homopentamer, and this is a functional 5-HT3 receptor. Okay, and we'll continue this discussion in the next video.